It is quite long. Little blurb about you. So just Ajay is known to everyone here, but I just like to say that he's a PI and MRC Career Development Fellow at Cambridge. He completed his PhD in Manchester with Professor Matt Landon-Ralph and is currently leading his lab in the MRC CBU with a focus on understanding diagnosis, prognosis in post-stroke aphasia using multimodal behavioral neuroimaging and computational modeling. Thank you.
Thanks so much, Fatima. I'm very excited and pleased to be a part of this symposium, and thank you all for coming. So I'm going to tell you about efficiency of aphasia assessment, and actually both the speakers previously touched on this, and I want to also make sure Blanca gets appropriate commendation here. So she led this project. I'm just going to get the pointer. So I'm going to whiz through the introduction very quickly because I think I'm preaching to the choir. And so we want to make sure that all of our assessments are accurate and reliable. And this is very important for things like differential diagnosis, figuring out where the breakdown is. And actually, it's very important, I think, for treatment planning and thinking about measuring recovery over time. It's not trivial, as our speakers before have mentioned. There's a wide range of deficits in stroke. And of course, it's not just in stroke, it's in many different disorders, ranging from memory, attention, language, even neuropsychiatric deficits as well. The severity is a big issue. It's a very heterogeneous population from mild to severe. Again, we touched on this in the previous talks. And so we have to make sure we are avoiding floor and ceiling artifacts in our assessments. And there's a trade-off between time. So of course, we would like to test patients for many, many hours, but sometimes it's not feasible. Some tests are designed to be more in-depth, and some are slightly more shallower and give you quick but relatively light information. And so detailed assessment in particular settings is just not feasible. So in clinical settings, in acute settings, we just cannot really assess participants for many, many hours and on many days. So the question becomes, well, how do we choose the tests that we want? And of course, this often is down to the specific questions of interest in the study. And we want to make sure that the tests that we are using actually get to the impairment that we're trying to measure. And so we want to have tests that are highly specific, but we want to also make sure, so we often refer to this as dynamic range, that there's enough graded variation in the test built in that we can grade participants along a continuum in more detail than just having a point score scale of 5 or 10. And as we talked about, the majority of tests, some are very quick for the acute setting. They take about 30 minutes to sort of an hour and a half, possibly. And many of these tests that I'm sure most of you are aware of, they cover lots of domains, but for each domain, there are relatively few a number of test items. And so we've been sort of thinking about this when we were developing our own programs. What was it that we really wanted out of the assessments, and what did we want to know about our patients? And my sort of main interest was aphasia. So if we think about aphasia is multidimensional. There's so many different aspects to it. And so when Matt and I started working on this, we sort of slightly joked around and said, well, okay, you can have different approaches. You can have the Rolls-Royce approach, which is, throw all the luxury at it, many hours of testing lots of different domains, in huge depth. You can, on the other end of the spectrum, you perhaps have the, this is called a Reliant Robin. Some of you may be familiar with this. It gets the job done, of course. It gets you from A to B. And in some cases, the test is shorter. Maybe they do test different domains, but as I mentioned, the number of items per domain is limited. We probably want something in the middle, maybe a reliable Toyota or similar. We want tests that can retain the sensitivity that we would get if we did the Rolls-Royce approach. And so that's where we started to really think about how do we sort of get to this middle ground. And so Matt and I spent a few years in Manchester building up an extensive battery on left hemisphere stroke aphasia cases. And we wanted to directly compare this with a popular shallow battery. So we know the CAT is widely used. And in the same context, we wanted to develop a novel battery which retained the sensitivity of the sort of extended battery that we had. So I'll go into this in a bit more detail. So in our group, we collect, it was actually about 15 hours worth of neuropsych, different aphasia and non-language tasks. Here are all the tasks listed on the side. I won't go through them in detail, but what we do is we apply a principal component analysis, and this table shows the loadings for each task with each of these latent components. And the idea was we could actually use this information to help determine which tests are most sensitive to tap into these latent dimensions. And you can do that by just looking at the loading. So the higher the loading, the more related these tests are to the component. But we want to avoid things where there's cross loadings, because that basically means each task loads across multiple domains, and it's not as pure as we might want to get. And so what we ended up with was a reduced set here. So you can see we started to sort of drop out a few tests. We had a pragmatic approach. So for example, you can see here for the repetition, there was a delayed version and an immediate version. Of course, the delayed version takes longer, so we decided to drop it. And we took another approach, which was actually to limit some taskers had a lot of items. We actually knew ourselves there were too many, and so we applied principal component analysis to the item level for each task. We get the loadings in the same way as you see here for each trial, and we just picked the top 50%. So these trials represent the trials that explain the most variance in the population that we had. So they're supposed to be the most sensitive trials. And so what we did was we dropped some of the tests by half, which again reduced the test time. And that's how we essentially ended up with what we call this reduced battery. It was driven by the data that we had for the extensive battery. And so this was published, I think, last year. So I'll just walk through very briefly the method. So we had 75 cases that completed the extensive battery. These were all based in Manchester. And we made sure that we recruited the full range of severity. So we had lots of severe cases as well as the mild and moderate ones as well. A subset of 40 completed the CAT, and I'm just showing the lesion distribution. We had controls also complete all tests so we can get normative sort of cutoffs. And in the paper, we do multiple analyses, but I'm just going to focus on two just for time. And so first thing we wanted to do was compare the sensitivity. So we essentially took a task from the extensive battery, and then we matched it with one of the tasks from the either the CAT or the reduced battery. And we wanted to see if they differed in sensitivity. And we're also interested in these sort of latent components, because the idea behind these latent components is they explain sort of independent information in the behavioral deficits. In this context, with the data that we had, we looked primarily at language components. And we wanted to make sure that the batteries were sensitive to these independent components. So this is how I'm going to display the results. Hopefully it's large enough for you all to see at the back. So we have the Palpa 9, which is just a word repetition task. We matched it with the cat's word repetition task. And here we've just got the percent that was marked as intact or in the normal range and in the impaired range. The correlation here, so the R squared here. But just the main thing to focus on is the last two columns. So what we calculated was, in effect, what proportion of participants were marked as intact with the cat, but were marked as impaired with a more extensive test. So in effect, the cat missed 15% of the participants because they were marked as intact, and 20% for the non-word. The same for some digit span, but I'll move on to the language one. So here's naming. So we took the Boston naming test. We have a Cambridge version as well. Again, it's about around 20% of cases missed. Changes a little bit more for the comprehension. So I've marked in red and blue. So in blue, there was just one test that we found the opposite. But the striking one that we found was synonym judgment, which is a very sensitive measure of semantic impairments. The cat really suffered in this particular domain. So this is with the extensive battery. So of course, you probably would expect this because this was about 15 hours worth of testing and the cat's about an hour, hour and a half. And so what we did was we now are comparing this with the reduced battery. So in the Palpa 9, we halved the number of trials. And again, we can still see the reduced, this novel battery, sorry, the cat missed 12.5% of the cases. Similar for naming, it was about sort of 10-15%, but now you can see there's slightly reduced sensitivity in the novel battery as well compared to the extensive battery. But still, for the comprehension one, we see this very high proportion of cases that were missed for comprehension sort of deficits. We wanted to see sort of who were these cases who were being sort of identified, and they essentially were the younger, more milder cases with smaller lesions, which probably sort of makes sense because these deficits are sort of harder to identify, and you need a more subtle and more in-depth testing. So here's sort of a figure version of the principal component analysis. So again, we had the 4 components. This is with the extensive battery. And I'll show you first the result from the reduced battery. So very pleasingly, we replicated the same structure. Again, this is using much less data than the full battery, but we find the same principal components, which is great. And this is the comparison with the cat. So here, what we have are all of the subtests of the cat. So there's the main language portion, and then there's a cognitive section as well. We didn't include the disability part here. But effectively, what this result shows is all of the subtests of the cat that are sort of language flavored all collapse into one principal component, and the executive ones collapse into a second principal component. I mean, essentially, I think what this tells us is there's not enough sensitivity in the individual tests to split apart some of these underlying like core language features that we can find if we have sort of much more detailed tests of language. And so, as I'm running out of time, I'll just go into the summary very quickly. So, as I sort of mentioned, we all understand that we need effective and efficient tools. How we do that is there's obviously lots of ideas. The previous 2 talkers, of course, have put together very nice tools. But one of the things that I think we really need to be very considerate of is the dynamic range. So the number of items in the test and the ability for those items to grade participants, because that actually will not only help us identify sort of mild deficits, but we can then use this type of scoring for, well, many kinds of analysis. But I'm very interested in longitudinal change. And of course, the more graded steps you have, you can be sure that you could be more sensitive to longitudinal change. So yeah, I've sort of repeated this. So yeah, there were some failings with the cat that we found compared to the extensive battery. But the key thing to note, which I didn't say before, is that we built this reduced battery so it took the same time as the cat. So I think that's the key point. So both batteries now take the exact same time. The extensive battery that we had took about 15 hours, which of course you would have to have a very luxurious setup to do that, and even in acute clinical settings, it just wouldn't be feasible. And so what we're moving on to next is we're really thinking more about harmonization across multiple sites. And so we're thinking about, well, how could we use the information from the extensive battery to help us sort of come up with tests that we can select that have a lot of items built into them. And so one idea that we took was, well, in the extreme limit, if you just take one task from each latent component from the reduced battery, so that's, the repetition has 40 items, synonym judgment, and there's a picture description task. This would give you a very good indication of the 3 core language components that we identified. You can extend this to the executive domain, and Rahel Schumacher has done that, lots of great work on that. And we're in conversation with Dorothy and Sungbom, who is here from Korea. And if you're interested, please do get in touch. I'm very happy to share all of the materials and information that we have. The very last thing before I close is, so we also built a bedside screener. So we use the same principle where we took the principal component analysis. We picked the top 10 loading items from all of these 3 tasks, so repetition, naming, and synonym judgment. And this can be completed in less than 5 minutes. We collected normative data, and we had a stopping rule that if they made more than 2 errors, then there's definitely evidence of some aphasia. So again, we're very happy to share that if you're interested. We're now currently implementing this in Addenbrooke's and trying to get clinical data from acute patients. So thanks to all the collaborators, thanks to our patients, funders, thanks to you.
Thank you very much, Ajay. We have time for questions.
Yes, Captain.
You want me to go first? Okay.
Advantage of being closer.
Thanks, AJ, for a really interesting talk. I just wondered, maybe you said it and I've missed it, how you chose the various tests that were in your battery, because arguably your executive tests, they're not tests that you would typically consider to be executive tests, things like the Raven, the Brixton, things like that.
Absolutely. So the first when we first built the battery, it was mainly focused on language and aphasia, and we included some quick executive tests because we wanted to have some non-verbal things built into it. And then when Rahel Schumacher joined the lab as a postdoc, actually she went to town. She collected loads of executive tests, all the ones that most people would consider as tests of executive function. And in her study, we found that you can then start to tease apart executive. So the component that we found previously started to tease apart into much more specific components of executive function. So in the current battery that we have, we actually have a much more extensive executive testing. I guess the idea really is, we want to be able to maximize the time that we have and make sure we get the most information we can get from the participants. The question is, how do we do that? And in the limit, Matt and I were discussing this. We were like, well, okay, you simply don't know the answer to, well, which tests are most sensitive unless you collect the data. And so we were in the position in Manchester where we built that huge battery, so we can then use that as our ground truth and say, look, if you spend 15 hours doing aphasia testing with some light sprinkling of non-verbal ones, then you can start going into that and unpicking it. So the question then becomes, okay, if you choose a different domain, you probably have to do something similar to essentially figure out, well, which ones are the best ones. I'm not sure how we get around that, other than sort of doing it. We've discussed it lots of times, but yeah.
Any more questions? Yes, Kathy, please.
Yes, yes, thank you, AJ. It's really useful to see how you can get the time down, do more items, but more efficient items. So that's fantastic. I also agree that it's really good to be able to pick up the milder phasics, so that particularly so you can do longitudinal studies, you can see them moving through the normal range. But I have the opposite concern about the cat, which is, we've recently conducted some online studies and normative data looking at their object naming scores. And what you see there is that there are plenty of normal subjects that, well, up to sort of 10%, depending on the item, who make the same sort of errors. So, when they're looking at a saxophone, they say clarinet, and when they're looking at pyramids, they say tense. So again, these are inefficient items in the cat that could be removed, but at the moment they're there, and they're obviously included in your analysis. But my concern is that we're picking up people who we think have anomia, but they don't necessarily have anomia at all, but it's the same as their pre-stroke level. So how do we know what's due to the stroke and what's not due to the stroke? And I think the same applies to some of the repetition tasks too. We assume that control subjects are going to, have optimal repetition, but if people lose their concentration for a moment, or they don't quite hear it, it's actually more surprising than with the naming tasks, that people can very easily make these errors, and that we're categorizing people as aphasic when they're not.
Yeah, no, I absolutely agree. I think it's always a challenge to figure out where the cutoffs are. Specifically, as you mentioned, controls will make mistakes, and so then we have to grade the patients in the context of we knowing that controls on average do make mistakes. So I agree that there is something about making, thinking more carefully about the trials that we select, whether we have to do this first pass screening with controls on lots of trials and say, okay, well, we're only going to pick the trials that are consistently, accurately, produced. And so I completely agree. I don't have a solution other than saying, I think what you've done is the right way to go. Collect lots of control data to basically see if you can start filtering out trials that are, more variable to begin with. But again, it's a way to, to maximize the in the analysis where we took the principal component analysis, when we took the principal components in the individual tests, that tells us which trials were most efficient at identifying some aphasia, but that was all done within the aphasia population. There was no control scores put in there. So that could be something that we could do as well.
Great, thank you so much. Thank you, Ajay.